Ladies and gentlemen, please welcome Stephanie Traub to the stage.
Good. Thanks for the invitation. I wanted to talk about today biomarkers, so the ideal biomarkers, the reality and future. So what ideal we want is that we have successful treatment for our patient. And then, from the industry perspective, we have our drug. And then, yes, we don't want to have adverse effects, no benefits for the patient, but have benefits as such, and design the ideal biomarker strategy to find that and make it happen. So, in fact, I always very struggling with that merge strategy. What does that actually mean? So I looked up the definition in the business directory, and actually it says it's a method or plan chosen to bring about a desired future, such as an achievement of a goal or solution to a problem, and the art of and science of planning and marshalling the resources you're having for the most efficient and effective use. So we have a method and a plan, and we have certain resources, and actually we want to use them to detect adverse events, identify the patients, monitor them, and identify efficacy, and on top of that, understand the disease biology. So the plan, the cunning plan in our biomarker strategy, so to boil it down, what does that actually mean? Well, we're asking a question. We sample at different time points. We measure and analyze with the different technologies. Then we generate numbers and answer the question, hopefully. So, some years ago, we did that pretty simple. So we took one biomarker and took that forward, what we call nowadays linear development. Then we thought, one biomarker is not describing the whole biology, so we need several to do signatures. So today we call that multifactorial development, so we need multiplexing. Then we thought, the multiplexing is also not doing the job because, in fact, we need the high sensitivity. So we took one again and tried to increase the sensitivity of the markers. And then we thought, that's also not doing the job. So we need multiple data sources. We need to take all that data and give them back to research and call that integrated development. So the ideal, to sum that up, is we need multiple data sourcing, we need machine learning, deep learning, what you've heard today, integration of omics data, big data, integration of signatures, integration of clinical data from competitors, from everywhere we can get them, and collaborate in big structures like IMI across industry to make that better. So, the reality. This is some tumor samples from one of our clinical trials. So this is tumor biopsies. What you immediately see on the right-hand side, there is one missing. That's actually quite often that the patient decline then to go to the torture again. So what you can see here, we have the stroma and matrix, we have immune cells. This is the marker we're interested in. So this is high throughput imaging analysis, so we know where our marker is expressed and to which level. So to cut that short, so you see that's another patient, very little tumor tissue, but this time we have a pre and post biopsy. Another tumor from another patient, even less tissue. Then, this is actually a bit better. So the pre one, we have some reasonable tissue. For the post, it's the same, so very difficult to get. And this is also a picture which is happening quite often. So here, in fact, you have normal tissue, what we got. There's actually very few tumor cells. So you see them encircled here. And then, as usual, the patient, or what's happening quite often, they decline for the second visit. And to not get you depressed entirely, that's a pretty good one. But if you see what's the difference, it's another tumor type. If you look like closely, so here we have like a good pre and post biopsy. So, oh, that was a tick. We asked a question, imagine a tick. We have sampled at different time points. We have measured and analyzed, and we have generated numbers and answered a question. So in fact, what you see here is the actual data, which I showed you in the slides before. So this is like the H-score of our markers. Here on the left-hand side, you see the pre and the post biopsy. And in fact, it doesn't say it comes from very little tissue. But what it would say probably in the clinical report is, well, we have like 4 samples. There's a slight increase in the marker X. There's like 1 outlier, which is going down. But in principle, there's a trend towards an increase of the marker, but we can't say for sure because actually we don't have enough samples. So, have we answered a question? Not really. So perhaps you want to say, well, tumor biomarkers from biopsies is very difficult. So let's go like in liquid biopsies. This is data we put together from CTCs, so that would be a liquid biopsy. We wouldn't have like all the problems with getting the biopsies and also like the several time points. I will talk you through the graph. So on that side, you see here like metastatic cancers, so small cell, prostate, breast, gastric. And here on the top is actually the number of CTCs you would find. So that ranges 1 to 3, 5, 10, 50, 100. And here the colors means like actually how many patients you would actually find. So what does that mean in that square? So in metastatic small cell lung cancer, 100% of your patients would have like 1 cell. Some of them would have like 2, then 80% 3, and then, well, perhaps around 60% would have like 50 CTCs. So what you can see, a lot of green, so that means like not for all of the specific tumor types. That would be like a real good measure. So there is some value probably in some of them, but I guess the CTCs unfortunately haven't brought us the solution to the measurement. Good, so what's with other liquid biopsies? Well, we have ctDNA, so circulating tumor DNA, and this is a really nice example, I think, where it's like working. So this is a DLBCL study. On the top, you see they have been followed with PET-CT, and here they got like the different treatments, and they followed them around for the ctDNA levels and also like the standard of care assessment to check for antibodies. And, well, so is this working like in all of the cases? This is data which have been published on the EGFR T750 mutation genotyping. And this paper, they did a comparison of ctDNA versus tumor. So in 30% of the test, they didn't find the actual mutation. So what does that mean is if the patients come out of the testing with the ctDNA and it's negative, they need to give a tumor biopsy to be certain if they have like the mutation and can get the treatment. So that's not that bad, yeah. So you would say, okay, both very difficult stuff to do. Here probably the sensitivity is missing. So let's go to something more simple, like blood or from blood derived serum and plasma samples. So what I wanted to show you here, so we have an initiative internally where we monitor sites for actually performance on the collection of samples. So here on the left-hand side, you see the number of deviations which occurred at that 5 different sites we're recruiting for the same study. In green, you see like the number of deviations per site. So you have to put that in context. That's a phase 1 study, and we have like recruited less than 20 patients, and they're getting around 6 to 8 samples taken. So actually that number is quite high. In gray, that's the number that affect the integrity of the sample. So to translate it and make it short, ideally you would take the sample and chuck it in a bin because there's like something went wrong and you can't use the sample. And you see here inside number 4, probably there's a big issue. In purple, you see number reportable in the final report. So that sample has like an impact on the analysis. You can still do the analysis, but in fact, you would need to make an asterisk on the sample in the final graph that something has happened. So what are all the deviations? So samples arrived too late at analyzing lab. So they have been degraded or they didn't get in time there. Volume less than expected. This is actually the blood tube, how it was filled. So the coagulant could be like different and influence your test. Outside the stipulated time frames, so either they took it too early or too late, which would fall then in that purple category because you can still use it, but it would need to be reported that actually that sample has been taken at another time. Incorrect number of aliquots obtained from sample processing. So we ask the people to aliquote the serum, and we would send like 10 tubes to lab A and 10 tubes to lab B. So they stuffed everything in one tube, so you can't send it somewhere. Good. Well, to sum it up, the reality. So samples taken might not be good quality. So in the context of the biopsies, perhaps they're like too difficult to take. And second, you have like the 2 shots. There are problems due to humans processing the samples, so at the clinical site actually. Sometimes the methods and technologies, they're like not sensitive enough for what we want to measure or detect reliable. I showed you the example of the EGFR mutation test with the ctDNA. The patients can't come in too often because, well, they have to travel. You have to take that in consideration that most of the time they feel like really poorly, so they don't want to travel. And also, like from ethics, well, you can't like siphon blood indeterminately. And the numbers I showed you, which are generated, are sometimes numbers which don't show the entire reality. I can reassure you the data you saw is like really what was measured. That was QA and QC checked, but it was like nowhere reported that actually the tumor quality was like not that good. Good. So perhaps we need to rethink and find like other ways how to solve all that problems. So I think we should like start an industry-wide initiative to improve how we take samples at clinical sites. The people are very busy there, so they have to do their day-to-day jobs, and then they take like samples for clinical trials on top of it. But you saw like how many deviations with actual samples have occurred, and probably that's a lot. Better, we get rid of all that sampling problem, and we do like real-time monitoring, but with required sensitivity and accuracy. Then we don't track like the patients like every 2 hours to the clinical site. So we do like home monitoring and give them like the chance that they can help us with the whole process. And we could do that with patient-centric sampling, like microsampling. They could take it at home, send it away, or go to their general practitioner. So immediately, biosensors, wearables, and new technologies spring into mind. Well, you might say perhaps they're actually not here yet, but I think in outside oncology, they are here already. It's like one of my favorite slides I always like to show. This is a tool which got like FDA approved 2 years ago. It's an implantable heart rate detector. And in fact, it's coupled to your mobile phone. And if it finds like inconsistencies with your heart rate, a nurse is calling you up and asking you if, well, you're feeling well. Yes, and what I also wanted to show you, that it's already here or going to be here. This is an article which just came out like actually last week when I read it. So the German parliament has like approved that you can get like health apps as prescription to follow around certain indications. So I think we need to follow in oncology. Thank you.